Right. So the basic idea here for the bacteriology course is that we're going to build on what you saw last year in the introduction to microbiology. So there's not going to be very much of repeating what we said last year. Okay, we're going to try and move on and cover material that is a bit more detailed. Okay. So what we have is we have 20 hours of lectures. That's 15 lectures, 9 hours of TD classes, and 3 lab sessions, which are 4 hours, 3 hours, and then 4 hours. And in all of our student evaluation of the module, everybody says the best part is the lab classes. Okay, that's the best. So what I have on Madoc for you is the slide sets for the lectures. Madame Huatou and Monsieur Deleva will be adding their slides later on during the semester. As you know, I try and record the lectures and put them up so you can listen to them again, because you all love the sound of my voice so much, right? Yeah. I'm also going to try and put written summaries, AI generated, but that I check afterwards, so that everybody has a kind of standard text version to go along with the lectures as well, okay? So that's experimental this year. Let's see how it goes. Now, the other thing that I like to do with ABT is microbe of the week. So once or twice a week after the lecture, I'll send you out a question by email at 7 p.m. in the evening. And you have to try and find the answer and email me back with the correct answer. And the idea is this is a kind of competition over the semester. So, it's kind of like points for the correct answer, bonus points for the person who writes in first. And yeah, so there's I think it's basically 3 points for the first correct answer, then 1 point every other correct answer. And the idea is it's kind of practice for you to find out some scientific information. It should be quite easy most of the time, but also try and write in English yourselves. It's really easy. You get to the Wikipedia page, you copy the section, you post it back to me. Oh, that's no good, right? So what I would like is for people to take the time and try and write out in your own words the answer. It's practice for you for your English, okay? Okay, so that will be starting this evening. I already sent the question out, but it will go out at 7 p.m. Okay, so everybody get ready for that if you want to play. So there's a kind of leaderboard, and at the end of the semester, my last lecture, there'll be prizes. Okay, there's a real prize every year. It's not just everybody gives you a clap and says you're great. Okay, there's a real prize here. Okay, so that's just for kind of like fun. It's optional, you don't have to do it. But okay, one good thing about this is, right, if you want to ask me for a recommendation letter in next year, I keep the records of this. So if you participate in Microbe of the Week, it gives me something positive to say, showed a real interest, took part in an optional exercise, gave very thoughtful answers, this kind of thing. I can write something positive about you if you play Microbe of the Week. Okay, so think ahead to next year a little bit. Okay, so for real evaluation, we stick with 60% on the exam and 40% CC, which is the lab report and the written test. Okay. Now, if you like microbiology, next year everybody gets to have 20 hours of virology and mycology. So that will kind of complement what we do this year, which is only concerning bacteria. And there is an option in semester 6, so basically about microorganisms, pathogenicity, and the microbiome. But you guys, you should all be taking the lamp placement if you can. Okay, in terms of other resources, so in the library we have these two microbiology textbooks, Prescott and Brock, and they're very useful. Often most of the stuff that we'll be presenting in the lectures is covered in these textbooks, and it's clearly written by someone else who will be explaining the same things in a different way. Okay, so if you can't understand something because, well, it's just the way I explain it, it just doesn't make sense to you, try and look in another source, okay, and see if it makes sense better that way. So they're good. And then these, websites, interesting just, for other stuff. Like this is a podcast. This is a kind of irregular blog from the group of people affiliated with the American Society for Microbiology. And these are kind of like images you can get of different things in microbiology. Okay, so back to the textbooks. So this is the program of what we're going to be covering during the lecture series. So it starts out with me. So I'll be doing the first 5 lectures. Structure of the bacterial cell, that will take more like 2 hours, not just today. And then the next 2 are kind of like bacterial physiology. And then 4 and 5, this is going to be energy metabolism, how do bacteria get energy, and some applications that are associated with that. Then François Deleva will be taking over for 2 lectures, and then Barbara Mouratou will come on and do something about regulation of expression, bacterial genetics, and then she comes back at the end with human microbiome, bacterial pathogens, and antibiotics. Okay, so I tried to note down where you find the similar kind of information in the textbooks. And I'm pretty sure for my own lectures it's correct, but for colleagues, this is kind of my best guess, all right? Okay, so why are we going to be spending so much time on bacteria this semester? Well, it's because they're everywhere. You don't notice it, but you've got bacteria all over this room, in our bodies, covering our skin, and also not just in kind of everyday environments like this, but also in extreme environments, down at the bottom of the sea, in hot springs, in a frozen lake underneath Antarctica. There are bacteria that are living there, okay? And they can do that because they have the ability to generate metabolic energy and live, pretty much on anything, any kind of favorable type of chemical reaction. Some bacteria somewhere will be able to exploit it and be able to live. And they can do this because they have a lot of fancy biochemistry that allows them to do things that, cells in our body just can't do. And that's very useful for industrial applications. Bacteria, depending on where they live, they will have enzymes that can work either at a very high temperature or a very low temperature. And this can be useful, okay? Another reason to study bacteria is, of course, because there are still a lot of very serious bacterial pathogens that infect and kill probably millions of people every year still. So we have to understand how they are hurting us in order to be able to, fight against them. Of course, most of the bacteria in our body are not harmful at all, but in fact are very useful for our, correct functioning, not only of our digestive system, but also our immune system. And there's a lot of links between the microbiome and neurodegenerative disease and things like that. Some things that are really quite surprising. So we're just starting to learn how important our commensal microorganisms are for our health. And the last reason is, well, bacteria are the simplest living organisms. And if you want to study the fundamental processes of life, then it's a good place to start. So all the big advances in understanding how DNA replicates, how DNA is expressed, how proteins are translated, all of these, or what the genetic code is, all of these advances were done using bacteria as a model system. And it's still pretty useful for that. Okay, so that's bacteria. They're great. They do lots of things. They're an important part of the living world. So let's just kind of refresh ourselves and refresh our memories about what a bacterial cell looks like. And it can be kind of like this. So in this kind of diagram, things that are in a dotted line, like this one and that one, they're facultative structures, okay? Not all bacteria have them. Whereas all bacteria have got to have, their DNA in some kind of nucleoid. They've got to have a plasma membrane. They must have a peptidoglycan cell wall. So these are underlined. These are absolutely essential. All the rest might be present, might not be present. It's okay. Absolutely essential for all bacteria. Some bacteria have an external membrane. Some have a capsule, a layer outside the external membrane. And bacteria may have different types of filaments on the outside of the cell. These ones kind of shorter and narrower, called fimbriae or pili, and a big flagellum here, which helps it swim around in the liquid environment. So what we're going to be doing for the rest of this afternoon and probably about the first half of the next lecture is to go through all of these bacterial cell structures. And the things I want, well, to understand myself, and I want you to understand, is what is the molecular structure of these different parts of a bacterial cell? What are the functions of these different parts of the bacterial cell? And how are these two things related? Okay, so it's the big thing in biology, structure, function, relationships. And what we'll do is start from the outside, look at the external structures first, and then the internal structures afterwards, which will be kind of quite simple. So we'll be spending most of our time on the external structures. So just like see if what you can remember from last year again, thinking about the bacterial envelope. Now let me just go back and start up the question here. Okay, start. Should be open. And you can talk about this with your neighbor. It doesn't have to be an individual test. Yeah. Ah, well, yeah. Lipopolysaccharide. Yeah, you're right. I should have spelled it out. Okay, everybody voted who wanted to vote. Yeah. Yeah, I hope everybody voted in July, right? Very important to vote. Yeah. That's why pensions are so high in France, because, if you look around Europe, France has got like the most generous pension system in Europe. It's because like old people, they vote, right? And you guys, you don't go to vote. Okay, so, right. Okay, so that's slightly off-topic digression there, I admit. Okay, so what did you say? So first answer that we need to look at here is what? Okay, the first thing I think is, C. What is C? Don't do that. Don't do that. Come back. Yeah. What is C? Because you need to understand that. So C is a lipid bilayer. So that's correct. So this is a membrane, this is a membrane, okay? B is a porine, yeah, that's right. Okay, this is a lipid membrane. B is a transmembrane protein that creates a pore. So molecules can cross the lipid membrane. Yes, B is a porine. And then 1 and 2, A is made of LPS. No, good guess, but no. And 1, it's the envelope of a gram-positive bacterium. No, it's false. It's gram-negative. Okay, why is it gram-negative? Because you have two membranes here, plasma membrane and the outer membrane. That's a gram-negative bacterium. With what in the middle? Not LPS, peptidoglycan. The cell wall made out of peptidoglycan. However, there is LPS on here, and it's these squiggly lines. Okay, the squiggly lines, that's LPS. Okay, so this thing is from last year, and this year you have to really become familiar with this and know it. So among eubacteria, I'm not talking about archaeobacteria, eubacteria, there are really two main types of envelope. And those are the envelopes of gram-positive bacteria and gram-negative bacteria. So gram-positive bacteria, they've just got a plasma membrane and then a thick layer of peptidoglycan, the cell wall. Whereas gram-negatives, they've got a plasma membrane, a thin layer of peptidoglycan, and an outer membrane. And this basic difference is the reason why they stain differentially with the gram staining technique. So gram staining works like this. First, and you're all going to do this in the lab and it will work perfectly. So first you fix the bacteria to a glass slide, then you stain them with crystal violet, which is a basic stain. It's going to just bind electrostatically to any polymer with a negative charge inside the bacteria. Anybody think what that might be? What kind of polymer do you have inside the cell with a negative charge? Okay, negative charge means it's going to be acidic, right? So something acid, it's in every kind of cell. One of them starts with an R, the other one starts with a D. Oh, yeah. Yeah. You have DNA and RNA. You have a lot of, they've got a lot of negative charge. So maybe the crystal violet is bound to these, RNA, DNA, and it will bind it will stain every single cell. And then you treat the cells with iodine, which is called the mordant, which forms crystals that can't easily leave the cell. And then you wash the cells with alcohol. So the alcohol will easily dissolve the outer membrane of our gram-negative bacteria, and it will wash out all of the crystal violet. Whereas the thick peptidoglycan layer in a gram-positive envelope resists alcohol pretty well. Okay, so gram-positive bacteria will not be de-stained with the alcohol wash. Okay, so at this point you've got gram-positives, they stay purple, and the gram-negatives become invisible, and that's why you have to counter-stain them with some other dye which will stain gram-negative cells pink. Okay, so one thing to remember: gram-positive purple, gram-negative pink. Now, there are some exceptions. Okay, so mycobacteria have a different type of envelope structure, and so they don't really stain gram-positive or gram-negative. You need a different type of staining protocol to stain mycobacteria. Okay, so let's have a closer look at the structure of these envelopes. So gram-positive bacteria, the structure is simpler. You've just got a thick layer of peptidoglycan, which is a kind of big polymer here, shown as these kind of like orange and yellow alternating blocks, which are linked together from time to time. So that's the peptidoglycan, and together with that you have another polymer called teichoic acid or lipoteichoic acid. And teichoic acid is a polymer of glycerol phosphate, or sometimes ribitol phosphate, ribitol 5-carbon sugar. And this kind of functional group here is going to be, I don't know, let's think, I have to read this. I think it's glucose or some kind of amino acid. Yeah, it can be glucose, D-alanine, or another amino acid. However, you have these long linear phosphoglycerol or phosphorybitol polymers, and what they do is they interact with different layers of the peptidoglycan by electrostatic interactions, and they just hold the whole thing together. Okay, some of them are anchored in the plasma membrane. These are the lipoteichoic acids, so they're anchored in the plasma membrane here, and they bind to the peptidoglycan up here. So their function is just to make sure the peptidoglycan stays attached to the plasma membrane or stays close to it. And the space in between the plasma membrane and the peptidoglycan, that's the periplasm. Okay, how about gram-negatives? So gram-negatives have a more complex structure. You have the plasma membrane, peptidoglycan layer, it's thinner, and an outer membrane. Now, the outer membrane has porins because otherwise soluble small hydrophilic molecules like glucose, amino acids would not be able to cross this membrane. Now, I forgot to say this, but this peptidoglycan layer is not absolutely, impermeable. Small molecules can diffuse freely across this peptidoglycan, okay? Whereas here, you have a lipid membrane, so small hydrophilic molecules cannot cross this membrane. So that's the function of the porins. Now, another thing that you have in gram-negative envelopes is what's called the Brown's lipoprotein. So it's got a protein part, it binds to the peptidoglycan, and it's got a lipid part that's inserted into the outer membrane. So its function is just to make sure the outer membrane stays attached to the peptidoglycan. It doesn't like float off somewhere. And the last thing that you have, which is specific to gram-negative bacteria, is that the outer leaflet of the outer membrane isn't formed of or made out of a regular phospholipid. It's made out of lipopolysaccharide, those squiggly lines that were in my previous diagram. So the outer leaflet of the outer membrane is made out of lipopolysaccharide. Okay, so we've got a couple of important polymers here, components, that we need to understand what their function is, how they're synthesized, and what their structure is, what their function is, and how they are synthesized. So the first one is peptidoglycan, the polymer that makes up the cell wall. Now, the cell wall is the rigid structure that's just outside the plasma membrane. And its function is to give a rigid structure to the cell and to protect against osmotic pressure. Everybody understands what I mean by osmotic pressure, right? Okay, so if you imagine you have a bacterial cell floating around with no peptidoglycan. The plasma membrane is very fluid, okay, it's like olive oil. And if you have water that diffuses inside the cell, it can easily just explode. The lipid membrane is not very rigid or stable. So the cell wall layer is really essential to keep a rigid, stable structure for the bacterial cell. Now, it's in eubacteria, the cell wall is composed of peptidoglycan, which is also called murine something. So, murine sometimes. And this structure is conserved among all eubacteria, so it's a target of antibacterial defenses. So lysozyme, which is an enzyme that we secrete. The big classic is, your eyes have got it, in tears. So any gram-positive bacteria that land on your eyes, they'll be attacked by lysozyme. It breaks down the peptidoglycan polymer. And the synthesis of peptidoglycan is inhibited by different families of antibiotics. For example, the beta-lactams, like penicillin. Also vancomycin and cephalosporins. Okay, so now there are a couple of exceptions. Okay, so archaeobacteria, archaea. Now they do have a cell wall layer because the function is essential, okay, keep a rigid structure to the bacterium, but it's not made of peptidoglycan. It's made of some other kind of polymer or sometimes just the S layer proteins. And another exception are the mycoplasmas. Okay, so the mycoplasmas are regular bacteria, eubacteria, and they don't have a cell wall. Now, I said that the cell wall is essential, so how do they survive? Well, they can only survive because they are parasites that live on the outside of eukaryotic cells. So what they do is they steal the cholesterol from the membrane of their host cell, and they use that to rigidify their own cell membrane. Okay, so they steal cholesterol, and that's why they can live without peptidoglycan. Okay, so everybody's going to need to concentrate pretty hard for the next few slides right now. Okay, so try and snap out of it over here and be concentrated. So peptidoglycan, what is the structure? So as its name suggests, you've got a peptide part and you've got a polysaccharide part, the glycan part, sugars and peptides. So the glycan part is what? It's a linear polymer of N-acetylglucosamine and N-acetylmuramic acid with a beta 1 to 4 linkage between them. So what do you need to remember about these things? Okay, they're both derived from glucose, N-acetylglucosamine. So the glucosamine, you're going to have an amine group on the number 6 carbon, and then an acetyl group on the top of that. N-acetylmuramic acid is slight modification of N-acetylglucosamine. Okay, so you have these modified sugars in a long polymer, so you go like NAM, NAG, NAM, NAG, NAM, NAG, you have a big linear polymer. And as well as that, the N-acetylmuramic acid, the NAM, has got 4 amino acids, a tetrapeptide attached to it, and the third amino acid is cross-linked to the fourth amino acid on another strand of the peptidoglycan. Now that's really important, because if you don't have the peptide part, what have you got? You've just got a load of linear threads of NAM, NAG, NAM, NAG, NAM, NAG. And if this is put under some kind of physical stress, it won't hold together, or it's just going to like blow apart, right? So the peptide cross-links, their function is to, turn this into something like some chain mail where all the links are holding the thing together. Okay, these strands are physically linked together, so they can't really be pulled apart. Okay, so that's what gives the strength to this structure. Okay, so what are the 4 amino acids in this tetrapeptide? The first one is L-alanine, the second is D-glutamine, the third is diaminopimelic acid, quite often, not always, and the fourth one is D-alanine. Now that's true in most gram-negative bacteria. In many gram-positives, the third amino acid will be lysine, and the cross-link won't be direct between the amino acid 3 and the D-alanine in position 4, but can be through a kind of glycine bridge. So that kind of makes sense, right? Because if you remember, in gram-negatives, the peptidoglycan layer is very thin. So the cross-links don't need to go across a big distance. Whereas in gram-positives, the peptidoglycan layer is very thick. So these cross-links can link together filaments that are further apart. So this cross-link, how does that work? What is important here is that both DAP and lysine, they've got a free amine group on the side chain of the amino acid. Okay, think back to biochemistry. In an amino acid, you've got the alpha carbon up here with a carboxylate group and an amine group. Same thing down here with lysine. So this is the side chain, la chaîne latérale. And on both of them, you've got a free amine group. And this amine group is going to form a peptide bond with the C-terminal carboxylate group on the D-alanine. Okay, so this thing here, this is a peptide bond between the amine group that's here and the carboxylate group on the end of the alanine. Okay, so now think a little bit about those structures and see what you can tell me. And once again, have a little chat about it. Everybody okay? Not very many votes. What do you mean? You don't care about amino acids? Well, you're on the wrong degree program then. Okay. Let's see what we got. Okay, just to kind of think about this. Okay, so this is what we put in. These are the answers that have come in. Peptide bonds are the wrong way round. That's not really. I don't. This doesn't really mean anything. This answer. So that was just kind of a fake one put in there to confuse you. But on the other hand, I can understand if you said that because you think, that you shouldn't have a peptide bond with the side chain. That's unusual. Okay, the cross link is not normal. So that's kind of, I can understand it. DAP, nobody thinks this is weird, right? For all of you in this room, this is just the most natural thing in the world. It's a regular molecule. You've all heard about it before, right? And there's nothing unusual about it at all. Come on, this is weird. This isn't one of the amino acids in proteins, right? Think about it. You've got alanine, glycine, valine, all these other ones. Nobody has diaminopimelic acid in their proteins. So this was one correct answer, right? Hey, there's something strange with this guy. Oh. And these other two, okay, one of them is correct, one of them is wrong. They can't both be right, okay? And the correct answer is answer 1, because the type of amino acids that are, that are incorporated into proteins are L amino acids. So it's not usual to find D amino acids in some kind of structure in the cell. So I think these two things, 1 and 3, these are telling you that this tetrapeptide isn't produced on the ribosome because it's the wrong kind of amino acids, D and a mixture of D and L, and it's got one amino acid that you don't find in proteins. So it's telling you that this whatever this biosynthetic pathway is, this isn't produced on a ribosome. Okay, so if that's not produced on a ribosome, how is peptidoglycan produced? There are basically 4 steps. So firstly, the repeating subunit, the NAM-NAG together with a pentapeptide is produced in the cytoplasm. Now you'll notice what's produced initially is a pentapeptide, whereas what you have in the mature peptidoglycan is a tetrapeptide. That's going to be important, so just keep that in mind. So the repeating subunits are synthesized in the cytoplasm, and then it's going to be flipped over with a lipid transporter called bactoprenol. And then there are 2 enzymes which are going to insert the subunit into the polymer. A transglycosidase, which will add this to the NAM-NAG chain, and a transpeptidase, which will form the peptide crosslink. Now, this is a complicated pathway, okay? And whenever you find something complicated in biology, well, I don't know about you, but me, I'm always asking myself, why does it have to be so complicated, okay? Wouldn't it be easier if it just did something else? Now, the reason this is complicated is because it's very difficult for a bacterium to produce a polymer outside the cell. Because these reactions here, producing a bond between 2 sugars, a glycosidic bond, or producing a peptide bond, they require energy to be input. But on the outside of the cell, okay, what do you use for energy in a cell normally, in a biochemical reaction? What kind of molecule is used by a cell to produce energy? ATP, yeah. Do you have any ATP outside the cell? Inside, yes, but outside? No, don't have any. So that's a problem, okay? How can you make a complex molecule when you don't have any ATP hanging around to push the reactions in the right way? So this is why it's using this complicated pathway, and we'll see that how it works in the next couple of slides. Now, the other thing that is interesting here is, the synthesis of peptidoglycan has got to be coupled with its degradation, because the mature peptidoglycan molecule is a very stable, rigid polymer. And if the cell wants to grow, okay, this is the cell growing, right? Then, of course, this polymer's got to be broken down, and you add in new subunits to be able to get the cell bigger. So when cells are growing, they're adding new subunits, but they're also breaking down peptidoglycan. And that's why when you treat growing cells with these antibiotic families that inhibit this pathway, these antibiotics are bactericidal, because the synthesis pathway will be inhibited, but the cells carry on destroying their own peptidoglycan by these enzymes that are called autolysins. So then afterwards, they explode, okay, because they've degraded their own peptidoglycan. Okay, so how does it work? Well, the first step is to produce this N-acetylmuramic acid together with the 5 amino acids of the pentapeptide, and it's linked together with UDP, which is uridine diphosphate. One thing to remember is the pentapeptide is attached to the NAM, okay? You've got NAM, NAG, NAM, NAG. Which one is it? It's the NAM. Why is it NAM? Because this is for N-acetylmuramic acid. Acid is a carboxylate group, which is on here. So this carboxylate group is where the first amino acid gets attached here with a peptide bond. So this is produced in the cytosol, and then it's going to be grafted onto the bactoprenol with a kind of pyrophosphate linkage here. So what's going to happen to this? It's going to become what? You have two, oh yeah. No. Okay, this is going to break off, okay? It's going to break off. Uridine diphosphate. So you have two choices. It's either going to stay UDP, or it's going to become UMP, diphosphate or monophosphate. Okay. When these two get joined together to produce this, who says you get UDP back? Nobody. Who says you get UMP? Yeah, you're right, yeah. So one of the phosphate groups from here stays here, right? So this is like spending one molecule of ATP when this happens, okay? So the energy here in that bond stays here with the N-acetylmuramic acid. Now this substance here is what's called lipid 1, and the last step to produce the repeating subunit is to add the N-acetylglucosamine, the NAG. So once you've got that, then you have your repeating subunit, NAG, NAM, and the pentapeptide. Now the next thing that happens is that the whole thing is going to be flipped over in the plasma membrane so that you have the NAG, NAM pentapeptide facing out in the periplasm. So this doesn't happen on its own. There's an enzyme that does this. Okay. Then once you have the NAG-NAM pentapeptide in the periplasm, the first thing that will happen is that there's a transglycosidase that will attach this NAM-NAG to a kind of free N-acetylglucosamine on the end of a filament of NAM-NAG, of peptidoglycan. So when this happens, where's the energy coming from to do this? Okay, the energy is coming from, you break this phosphodiester bond here, and that releases the energy that allows you to make the new glycosidic bond. Okay, so that's how it can work. You break one bond, you make a new bond. It's neutral. Okay, so the energy that got put in with the UDP, that's what it's used for. You cash in on this step. Now, the bactoprenol will be recycled, flipped over back in the plasma membrane, so it's ready to start another round in the synthesis. And the pyrophosphate will be cleaved off onto the monophosphoryl form. And the last step is the formation of the peptide link with the transpeptidase. So what is going to happen here is that the transpeptidase will form a peptide bond between this free amine group and the C-terminal part of the fourth amino acid, D-alanine. So if you imagine this one, the last two amino acids here, you've got a peptide bond, and what's going to happen is this peptide bond is going to break, so that's going to release energy, and you use that energy to create the new peptide bond here. So you break one peptide bond, and you make a new one. That's free. It's neutral. It's at equilibrium. And this will have one molecule of alanine as a leaving group, which will be re-imported into the cell. So that's why bacteria have to produce the precursor as the NAG-NAM pentapeptide, because the fifth amino acid has got the peptide bond, and that peptide bond contains the energy for the transpeptidase to work. So that's how they do this. It's a wonderful trick to be able to form a large complex molecule outside the cell where you don't have any ATP. Okay, let's just think about this one. Yeah, I think we should do this. See if you understood this. Oh yeah, this is where I realized I didn't tell you one essential piece of information. But that's what the question is there for anyway, to remind me. Okay, let's see what we got here. Right, so okay, well, we've got kind of a mix here. Okay, 4, almost nobody took 4, that's good. 4 is wrong because we're talking about gram-positives. Gram-positive bacteria, they don't have porins. So like out of the equation here. So 4 is wrong. 5 is also wrong. It's like, you know, if you're starving, why don't you just evolve a way not to need to eat food? And you'll be fine, right? Come on, this is almost practically impossible, right, for bacteria. There's not really a practical solution right here. 1, express an enzyme that destroys the antibiotic. Yes, that's a mechanism of penicillin resistance. Beta-lactamase is the enzyme which can destroy penicillin and other enzymes of the same family, other antibiotics of the same family, excuse me. So 1 is correct answer, and out of 2 and 3, 1 of these is correct. It's answer 2, because what I didn't say is that these beta-lactam antibiotics and cephalosporins and vancomycins, they all inhibit the transpeptidase activity. Okay, so they are all acting at the last step in the synthesis of peptidoglycan. So these are the molecule, well, transpeptidase, well, not vancomycin to be honest, but beta-lactam antibiotics will bind to the transpeptidase and inhibit it. So if the transpeptidase mutates so that it doesn't bind the antibiotic anymore, that bacterium will become resistant. Okay, so 2 is correct. Okay, so that's peptidoglycan. So our next molecule that we have to understand is LPS, lipopolysaccharide. Now, lipopolysaccharide, once again, the name is giving you a bit of a clue here. You have a lipid part and a polysaccharide part. So the lipid part is this thing down here, and it's not a normal lipid, okay? It's got 1, 2, 3, 4, 5, 6 acyl chains, fatty acid chains, connected to 2 kind of weird looking sugars up here. So this thing is called lipid A, and it's only found in the outer membrane of gram-negative bacteria. Very, very specific to gram-negative bacteria. Now, going up from the lipid A, you have the polysaccharide part. So first there's a core oligosaccharide. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 sugars that form this structure. Starting out with some weird things like heptulose and this other thing called KDO, which I imagine you have never heard of. Once again, this is only present in lipopolysaccharide, in LPS. Now, the core oligosaccharide, it's kind of short, it's compact, and then the last part are the O antigen repeats. So this is the polysaccharide part. So this is composed of short motifs, 3 to 8 sugars, but they're repeated 10 to 40 times. So you have the lipid part and then a big, long repeating motif of polysaccharide. So what's the function of this thing? Well, its main function for bacteria is to protect the bacterium against chemical assault, either biosalts, detergents, things that can attack the membrane. And so it's got a function of protection. Now, because the other thing I want to say about this is that as you go from the lipid part to the O repeats, the molecule becomes less. Yeah, I should talk about it. I don't know. Okay, I'll carry on with what I'm saying, and I'll come back to this later. No, I should do this first and then do. All right, just imagine that the last 2 minutes never happened, okay? So, function for the bacterial cell is protection. However, LPS is a conserved molecule, and it's specific for gram-negative bacteria. And there are receptors in our immune system that can detect the presence of LPS and induce inflammation. So that's part of our immune response, and the presence of LPS can cause fever, diarrhea, and in the worst cases, septic shock, and even kill you if you've got a very severe gram-negative septicemia. Now, because of these harmful effects, lipopolysaccharide is also called endotoxin because it has a toxic effect on us, okay? And it's endotoxin because it stays associated with the bacterial cell. It's not something that's secreted out of the bacterium. It's part of the bacterial envelope. So that's the effects on the on health. And this thing, the repeats up here are what's called the O antigen. O, it's sugar, it's an O's, okay, which is useful for the identification of different strains of bacteria. So why is it the O antigen and that is the useful part for identification? Well, it's because as you go from the base up to the tip of LPS, the molecule becomes less and less conserved. So lipid A, that's identical in all gram-negative bacteria. So the core oligosaccharide, that's going to be the same in different species from the same genus or the same family of bacteria. You have some diversity within the whole of gram-negatives. But once you get up to the O antigen, then this is very, very variable, even within the same species. So if you want to identify a strain of bacteria within a species, you can use the O antigen because you'll have, 10 to 100 different O serotypes within the same species. So how is this thing synthesized? It's the same kind of problem for the bacterial cell. It's on the outside of the cell and it's a complex molecule. So how is it synthesized? Well, the core lipid and the O repeats are synthesized separately. So the core oligosaccharide is synthesized in the cytoplasm attached to lipid A. Then this is flipped over into the periplasm. Whereas the O antigen subunits, they're once again synthesized in the cytoplasm, attached to some kind of lipid molecule here, flipped over, and then you have a transglycosidase, which will add on the subunits to a long chain. And then after a certain number of repeats, then the whole of this chain is transferred over onto the core. So then you have a mature LPS molecule, which is in the wrong place. And the last step is just to take this whole thing and shift it up to the outside of the cell. Okay, so any ideas what this might be here? So if I'm asking you the question, it means you might be able to guess it from what we've just seen. What could it be? Okay, so in general terms, what is happening here is something very similar to what we saw for peptidoglycan. You have a repeating subunit of some sugars. It's flipped over in the membrane, and then a transglycosidase makes a big, big polymer. So what was this attached to for peptidoglycan? Flip back and check. Bactoprenol, okay? So it's the same thing. This is bactoprenol here with a diphosphate, pyrophosphate linkage, which is doing the same thing. So the bacterium has got the same kind of problem, how to form a polymer on the outside of the cell, and it's using the same solution. Okay, it's doing the same kind of thing. It's not inventing a totally different biosynthetic pathway. Okay, capsule antigens. Another thing we need to know. So these are, facultative structures that are really the outermost layer of a bacterium, not always present, but can be present in gram-positives and in gram-negatives. Now, when a capsule is present, it can often be seen by light microscopy. You can see a kind of little diffuse layer around the outside of the bacterial cell and can easily be seen on electron microscopy here. Now the function, okay, so it's not an essential component. Not all bacteria have a capsule, but when it's present, it can be used to identify bacteria again, okay, present or absent, and there are different serotypes. So this is called the K antigen, K for capsule in German. And the function of the capsule antigen is once again to protect the bacterium against detergents, antibiotics. It's forming a big impenetrable layer here, so things can't really get through. Bacteriophages, viruses that can attach to bacteria and kill them. Sometimes they can't get through the capsule layer very easily. They can protect against desiccation, loss of water. You know, if bacteria are growing on a surface and it dries out, they can just dry out and die. But the capsule polymer is often very hygroscopic, so it retains water. And, you know, it's slimy. The bacteria keep hydrated even if all the water's gone. And last thing is, often the capsule can be important virulence factors for bacteria, for bacterial pathogens, because they protect the bacterium against phagocytosis. Okay, so what are they made of? They are polymers, mostly polysaccharides, sometimes polypeptides on the outside of the cell. So how are they produced? Well, it's the same kind of thing. It's similar to the LPS-O antigen, basically. So you've got synthesis of a repeating subunit on bactoprenol, and then you'll have the bactoprenol and the subunit, which is translocated to the periplasm. Then you have a transglycosidase activity, which will form the polymer. And then at the end, you've got a big multi-enzyme complex that will pick up the whole thing and push it out to the outside of the cell. So once you've understood how peptidoglycan is synthesized, you can really kind of understand how LPS and the capsule antigens are formed, because it's the same kind of problem and the process is very, very similar. Okay, what else have we got on the outside of the bacterial cell? We have fimbriae and pili, which are two different names for the same kind of structures. So fimbriae and pili are these like very short filaments that you can see covering the whole of this bacterium here. Okay, you can see them come out here. So they can be very numerous. You can have like 1,000 per cell. And they're very fine, very thin, 3 to 10 nanometers in diameter, and they can have multiple functions. So most of them, or many types of pili, are essential for adhesion, either to, you know, the surface of a rock or piece of plant material. And they can also be involved in adhesion to the mucosa of a host organism. So they can be important for invading hosts. So bacterial pathogens can have pili that are the adhesins that allow them to invade the host. Some of them are kind of like needles, which are, you know, hollow, and they can inject stuff or export proteins. So they can secrete virulence factors, and others can be used to move along a surface. Now, as well as that, you have conjugation pili, which are less numerous, 1 to 10 per cell on the slide, often maybe 1 or 2. Now, these are slightly wider diameter, and they're essential for the conjugation between bacteria. So conjugation is the exchange of DNA between two bacterial cells. So what will happen is that the bacterium that expresses the F. pilus, or pilum, I guess, pilus, will extrude it, send it out like a harpoon. It will bind to the neighboring cell, and then this bacterium will kind of reel it in like this. So this filament here will shorten, and the two bacteria will end up stuck one next to the other one. And then when they're together, that's when the exchange of DNA can occur, from the bacterium which is F plus to the bacterium which is F minus. Okay, so two names for these filaments. So why do we have two names? It's because they were discovered around about the same time by two microbiologists, one in Scotland called Mr. Duguid, and he called them fimbriae, which is like a thread or something in Latin. And there was an American microbiologist around about the same time described the same structures or similar structures, and he called them pili, like for, poil, hairs on the outside of bacteria. So that's why you've got two names. Really, it's the same thing. So I'm just going to talk about pili now, okay? So I'm just going to use that name. Now, as well as having two names, there are different types of pili. And, as you can see, there's quite a lot of different functions here. Adhesion, motility, transfer of DNA, secretion, different things that they're doing, and they're not all the same. So there have been different ways of classifying pili over the years. So the first one was proposed by Monsieur Duguid, or Duguid maybe. Duguid. So his classification was based on the biochemical properties of some of the pili. And then the ones that he characterized, they had these kind of biochemical properties. And then he had like other categories for the conjugation pili. He put them apart. These are the F pili. And then 5 and 6, pili present in other bacteria that didn't really kind of fit with the classification scheme. So then more and more people began finding different types of pili. So 10 years later, Mr. Otto decided that they would reform the classification. And he said, like, okay, for all of Duguid's 1, 2, 3, 4, they're doing the same thing. They're all adhesion pili. So I'm going to put them all in the same class. Conjugation pili, they're doing something else. That's class 2, the conjugation pili. And then other groups for pili in different bacteria where we didn't really know what was going on. Okay, or this one, number 4, that was responsible for gliding motility. So the classification of Otto is more based on function. Now, as we've begun to understand more and more about the structure of different types of pili and how they're synthesized, the modern classification is really based on their molecular structure, okay? So this group of adhesion pili was expanded to like the Cu and the curly pili. This gliding motility, that's something that's typical of type 4 pili. And these ones, Agrobacterium, these are type 4 secretion pili, and other pili involved in secretion are type 3. Conjugation pili become F pili again, and within the last 10 years, there's another group that's been discovered. So this is the current classification. And sometimes we still talk about type 1 pili because that's a subgroup of the Cu pili. Okay, so that's the current classification, and the different types do different things. So Cu, curly, and type 5, they're only able to adhere to surfaces. That's it, that's their function. 3 and 4, secretion, or the 4 secretion ones. And then the 4, some of them can be used for motility and adhesion. So the structural groups kind of fit with the functions. So why is that? So there's a rather complicated slide, I'm afraid, to illustrate the structures of different types of pili in gram-negative bacteria. So types Cu, curly, and type 5, their function is adhesion only. And what they have in common is that they're just anchored in the outer membrane of the bacterium. Okay, somehow bacteria produce these things, they're anchored in the outer membrane. Once they're there, the bacterium can't do anything else, okay? They're just involved in adhesion, that's it. Type 3 secretion pili. Okay, the structure is complex. You have several proteins forming this structure that crosses the plasma membrane and the outer membrane. And then you have a filament which is hollow, okay? So it's a kind of spiral of protein and the inside is empty. And this can be used to secrete proteins that are excreted from the bacterial cell to the outside. Now the filament here is produced by, well, it's formed of hundreds of copies of one filament protein. And the filament protein is synthesized on the ribosome, and then it's transported up the inside of this structure. It goes all the way up to the filament and adds onto the top. Okay, so type 4 pili, they're a bit different. So type 4 secretion pili. Similar kind of structure, the big protein complex that crosses the plasma membrane and the outer membrane, and a hollow filament that allows proteins to be transported from the cytosol and secreted outside the cell. But the big difference between type 4 pili and type 3 pili is that the filament proteins that form this hollow tube, they're added at the base of the filament here, okay? So that's true for type 4 secretion pili and also true for the type 4 pili that are involved in gliding motility. So what happens here is that these type 4 pili for moving around are able to do so because the addition, the polymerization occurs down here at the base. And because it's in the plasma membrane, the bacterium can control what's going on. So this process is going to take ATP, but it can be reversed. Okay, so that means that type 4 pili can be extruded and retracted pretty much as the bacterium wants. That's also the case for the type 4 secretion pili. And in fact, that's also true for F pili, as we saw, the conjugation pili. So conjugation pili, they're a kind of modified form of the type 4 secretion pili. They're hollow, DNA can pass through the tube, and they can be extruded and retracted. So just to illustrate this, and then after I've done this, it will be a good time to stop, I guess, because we're out of time. So gliding motility using type 4 pili, how does it work? Well, you start off with this bacterium that's on a surface, and the type 4 pili are attaching it to the surface. They have an adhesion function as well. So the first thing that's going to happen is this bacterium will extrude new pili at the front. Then it will kind of retract the pili that are at the back. So it's not attached here anymore. Then the extremity of this pilus, the distal end, is going to attach onto the surface here with its adhesion function. And then once again, these pili will be retracted, and that generates force for the bacterium to move forward. So it's only type 4 pili that can do this, because this is the only type of pili where the polymerization, that is making the filament longer, and the depolymerization, making it shorter, can be controlled by the bacterium. Okay, so that's why only the type 4 pili can do this. Okay, so I think that's about a good place to stop. So I'll stop here. Don't forget, 7 p.m. the question is going to be sent out for Microbe of the Week.